We speak student.
Molecular genetics, RNA. A la Shmoop. We are here with Dr. Ruth Tenen to talk about molecular genetics here at Shmoop Global Headquarters in Mountain View, California. And we're going to cover genetic code and DNA, RNA structure. All right, so moving right along, what is RNA? We've talked about DNA. What is ribonucleic acid?
Yes, so it's very similar to DNA, but it has that extra oxygen. So it's also a polymer of nucleotides. One big difference is rather than the thymine as one of the bases, it's uracil. So T goes to U.
Got it.
They're basically the same. Otherwise, their function's very different, and there's other differences as well. So, for example, DNA is always double-stranded. RNA tends to be single-stranded and then fold back on itself. So they have different functions in the cells, but they structurally are pretty similar.
Got it.
RNA.
Okay. Then we have subsets of RNA, mRNA, rRNA, tRNA. Can you explain to us, like, what are those elements and how do they function? What are the different kinds of RNA?
So kind of the major RNA that we always hear about is mRNA, which is the intermediate between DNA and protein. So DNA gets transcribed into mRNA, which gets translated into protein. So that's kind of like the famous RNA. But there's what's called non-coding RNAs that aren't turned into proteins, basically. So tRNAs are transfer RNAs, and those are involved in protein synthesis. They help with the ribosome. There are ribosomal RNAs, which again, they don't get turned into proteins, but they actually function as RNAs.
But walk us through, like, how does it work? Like, what's the RNA process? Why do we need these interstitial steps to make things happen so that everything's okay? Is it like a human checking mechanism to be sure the genes are all happy and healthy, and if they don't, like, the cell goes away so we heal properly? Why do we need RNA?
One reason is that DNA, we want it to be super stable, right? Because it gets passed down through all the generations and we need to keep it the same.
Sure.
By having an intermediate, basically you can say, okay, I want to turn on this gene and I'm going to make a ton of copies of the messenger RNA. So you can have a ton of different copies that ultimately will get degraded when they're not needed anymore, whereas you wouldn't want to chew up the DNA when it wasn't needed anymore. So it's a way of...
How do they get degraded? So this is like taking a DVD copy and like copying it again and again and again, and after a while it just, like, the data isn't as clear?
So each copy actually is originally from the DNA. So each copy is just as good as the previous one, but eventually, let's say the cell wanted to turn on a gene to do a certain thing. So it said, okay, I'm gonna need to metabolize sugar right now, so I gotta turn on my sugar metabolizing gene. And then the sugar goes away, it doesn't need that protein anymore that would do that. So then there are enzymes that come in and they'll just chew up and get rid of the RNA, the extra.
Got it. So it degrades not in that its quality gets less, it degrades in that its utility is lower.
Exactly,
Meaning,
the abundance ends up going down when it's not needed anymore.
got it. And our body has stuff that regulates that? Is that like the
I
things
don't
in your
know
brain?
in terms of actual hormones and things, but at the cellular level, there's like a million enzymes that are always plugging around. And basically, these processes are super important, so there's tons of layers of regulation just to make sure everything's very tightly constrained.
Got it, because you got a lot of replications happening. I mean, it kind of reminds me of a computer. It's like if you don't have lots of checks in your database, you're going to print bad data. What is RNA? What are the different kinds of RNA? Why do we need RNA?